Since the birth of organ transplantation, there has always been an unmet clinical need from the shortage of organ donors, with 8,000 annual waiting list deaths. What if, using your own stem cells, you could print yourself an organ? Or more accurately, bioprint yourself an organ. But what is 3D bioprinting? 3D bioprinting is a technology that is capable of distributing cells and biomaterials in a precise fashion that is capable of mimicking the internal environment of tissues in vitro, which could be ultimately used to create organs for transplantation in vivo. How does it work? Regardless of the desired tissue type, there are 4 main steps involved. Let's take a closer look at each step. Step 1. Choose your bioink. These are natural or synthetic polymers which will form the base or scaffold of the bioprint. Step 2. Print the scaffolds. Scaffolds are printed using a 3D printer and the selected bioink. Step 3. Incorporate stem cells. Stem cells are incorporated into the scaffolds, which serve as a guide by providing optimal conditions for stem cell differentiation into specialized cells and ultimately tissues. Step 4. Differentiation. Altering the environment under which the stem cells undergo differentiation will determine the tissue type. Factors like temperature, UV, and pH are all used to create the desired tissue. Different growth factors can also be introduced, including vascular endothelial growth factor, transforming growth factor beta, and fibroblast growth factor, which will also alter the morphology of the final tissue. Stem cells are the primary instrument in bioprinting. Different stem cells can be used, however, induced pluripotent stem cells, or iPSCs, are the most favourable due to their outstanding pluripotency. iPSCs are derived from the skin or blood and using transcription factors are reprogrammed back into an embryonic-like pluripotent state that enables the development into any human cell. As they're derived from own cells, there is also a much less risk of an immune response compared to receiving an organ donation. Different bioinks in isolation or combination are better suited for printing tissues with certain properties. For example, collagen is a primary component of the extracellular matrix. This makes it a good choice for printing high-power tissues like cardiac muscle. Contrastingly, alginate is a much softer bioink. However, with the addition of gelatin, it can be used to print scaffolds for tough tissues. Or, using a combination of alginate and methylcellulose will result in high shape fidelity, which could allow for modification of tissues post-printing. Hydrogels are clusters of polymer chains that can mimic the microenvironment of tissues in vitro, which increases cell viability and promotes tissue generation within bioprinted constructs. Printed constructs require vasculature to prevent necrosis. Bioinks can be used to print organs with pre-formed vasculature to be perfused immediately to prevent cell death in the printed tissue. There has been success seen within bioprinting a range of tissue types, including connective, muscle, epithelial, and nervous tissue. Most of the attention, however, is on muscle and nervous tissue, as they're both tissue types with very little regeneration capabilities. Let's first take a look at muscle tissue, but more specifically, the heart. Cardiac tissue has very little regenerative capacity, which is one of the reasons that cardiovascular disease is and remains the leading cause of death globally. iPSCs have also been used to bioprint cardiomyocytes, which are responsible for contraction of the heart. Cardiomyocytes can sense the direction of bioprinted scaffolds, which ensures accurate spatial distribution. Incorrect organization of cardiomyocytes results in a multidirectional contraction, which results in an overall course of 0. Some scientists have used iPSC-derived cardiomyocytes to bioprint beating cardiac constructs, with the beating becoming synchronous 7 days post-printing. Drugs that affect native cardiac tissue function cause similar effects in the printed constructs. For example, isoproterenol administration resulted in an increased heart rate, as you would see in vivo. Just like the heart, nervous tissue also has very little regeneration potential. More specifically, the spinal cord, which is also a prominent area of interest within bioprinting. The complex structure of the spinal cord, especially the electrical conductivity, has halted progress in developing treatment of spinal cord injury. Scientists have also used iPSCs to create neural progenitor cells. When incorporated into a bioprinted scaffold, neural progenitor cells undergo axonal propagation, creating a living platform. This living platform is biomimetic, which means it is capable of communication through electrical signals, just like the spinal cord. Researchers have used these living platforms in mice, which have been given spinal cord injury. The living platform is injected into the cystic cavity of the spinal cord. This induces regeneration of neurons at the active site of the injury, which results in mice who were previously unable to walk to regain their ability. 3D bioprinting has opened up new frontiers in the field of regenerative medicine, but as with any new technology, it comes with its own set of challenges. While 3D bioprinted tissues have shown promise in early studies, the long-term effects of exposure to these tissues remain unknown. Another challenge is the risk of tumour formation due to the outstanding pluripotency of iPSCs. Additionally, replicating organ failure is a major concern when using autologous stem cells. This is because the stem cells used to create the 3D bioprinted organs may have already been compromised by the underlying disease or condition that caused the organ failure in the first place. Finally, there is the issue of unknown variability in the printed tissue, with no legal and ethical way of testing constructs. This means that there is currently no way to ensure that 3D bioprinted tissues are consistent and safe for use in human patients. Despite these challenges, scientists and researchers continue to work towards refining 3D bioprinting technology and overcoming these obstacles. With continued research and development, 3D bioprinting has the potential to revolutionise regenerative medicine and bring hope to millions of patients in need.